Okay, so hi. So this is section 2 of lecture 1. So we have just finished section 1 where, to recap, we were discussing the Streptococcus pneumonia bacteria. So Streptococcus pneumonia is a bacteria that causes pneumonia, and part of the mechanisms it uses to become pathogenic is this quorum signaling pathway, where it expresses from one of its genes, the COMP gene, this small peptide, competent stimulating peptide, which it then has a receptor for, which then activates its positive feedback, the higher expression of itself. But it also turns on downstream pathogenic genes, such as genes which cause it to go into a biofilm, a film of bacteria, which makes it more resistant to antiseptics, for instance, causes it to become competent, which means that it actually absorbs genes from other streptococcus bacteria, which is particularly bad because they may contain drug resistance genes, which makes it more sort of resistant to antibiotics. So, so the idea of showing you this system was really to give you an idea of the biological system composed of biological networks, and also omics data, how we can interrogate the system using omics data, um, and machine learning essentially. This was an example of clustering data over time, and also genomics data of the organism. And so this was an example of pattern recognition, of cis-regulatory motifs on the controlling the genes involved. So this was really just I didn't expect you to fully understand the detail. It was really to give you a sense of what the whole idea of systems biology is about and applying machine learning. To give you a more formal introduction to what we'll cover in this lecture, so learning outcomes. I'm hoping by the end of the lecture you'll have an idea that how biological systems can be understood in terms of the parts of the system and the interaction between those parts. You'll be able to outline the key types of biological networks involved in the cell and how they work in terms of the nodes and the vertices of those networks, and describe how omics high throughput technologies work and how they relate to understanding the biological systems. All of this is a natural domain for machine learning, as I hope this sort of first example indicated. So there are four main types of biological networks within cells, and I wanted to show how this works in terms of a biological process that you might be familiar with. So imagine an insulin hormone stimulating a liver cell to turn on the processing of glucose to glycogen to reduce the blood glucose level. So you might be aware that type 2 diabetes cells do not respond so well to insulin, and this causes sort of the lack of control of blood sugar, that the blood sugar can become too high. How does that work? Well, the insulin comes along and binds to an insulin receptor on the cell surface. This causes a signaling pathway cascade, so phosphorylation of particular proteins causing the phosphorylation of other proteins and other processes, and this takes this signal that insulin is bound all the way down to the nucleus. Then we have a different type of pathway, gene regulation pathways, This is where one gene might be activated and expressed, so its activity goes up, and it might produce a transcription factor protein, which then turns on another 10 genes, and then one of them might turn off 4 or 5 genes. And what we have is a cascade of genes all being turned on and off due to the binding of the insulin. So this is gene regulation, and this in turn, if 20 of these genes are enzymes, it may end up turning on, say, 10 enzymes and turning off another 10 enzymes. So we've got the metabolic pathways where enzymes basically process metabolites. So it might process glucose into glycogen, and it may stop processing glycogen into glucose. So that's the metabolic pathways. These are all pathways which are real biological parts of the cell, essentially. They're interacting parts of the cell, interacting molecules. There is a fourth type, protein-protein interaction networks, which is not really a pathway. It's because the experimental techniques allow us to measure which proteins interact with which other proteins. So we've built up databases of proteins interacting with proteins just because of the experimental technique. These are involved in all of these processes. So for the receptor to phosphorylate another protein, we will have to have protein-protein interactions. A transcription factor might bind to another transcription protein to actually turn on other genes. And metabolic pathways may consist of enzymes which bind onto each other so that metabolites can efficiently be processed. So these are within the liver cell, responding to insulin. These are how the 4 main classes of pathways and networks. I think it's good to go back and compare to the quorum sensing mechanism. Again, we had a different type of hormone, a small peptide pheromone binding just to a bacteria. So there was no nucleus or anything, it's just a bacteria. But it did turn on the signaling pathway, which then turned on gene regulation pathways to actually turn on and off quite a number of genes, which we saw in the microarray gene expression data, and we could find the motifs of the binding transcription factors. So biology, there's this evolved consistency about the detail is different between different organisms, but a lot of the same processes are going on. So to give you even more detail, so this is clearly not something that you need to remember or know about, or I really want to just show you this to give you the sense of how complicated biological systems are. I started with the quorum mechanism for Streptococcus because as a bacteria we can sort of understand most of the parts and how they're working. Whereas eukaryotic cells like the liver, it's sort of incredibly complex to understand. So what do we have here? Well, we have more detail of the insulin receptor, the IGF-1 receptor, insulin growth factor 1 receptor. So insulin is binding here, and it causes this, so it's transmembrane, it's going through the lipid membrane, and it's causing self-phosphorylation of this receptor. And you can sort of see that this is causing a number of pathways to be activated in systems biology networks, we usually indicate activation using an arrow and then repression of a process using a bar like this. So we've got a protein here that is repressing both this and this, but this is causing these IRS proteins to become phosphorylated, which in turn is activating RAS and these MAP kinase proteins. And this is a cascade of kinase which leads to sort of gene expression. And then we have this being activated, phosphorylated, which binds to these proteins and causes glucose transporters to go from a vesicle inside the cell onto the surface. So it increases the number of glucose transporters coming in to this, taking glucose into the cell. This phosphorylation also causes this this pathway involving phosphatidylinosole 3 kinase to be activated. Both of these leading to cell growth, and these leading to glucose metabolism, glycogen lipid synthesis. So if it's a fat cell, insulin causes the absorption of glucose, but also that turning that glucose into lipid. Whereas if it's a liver cell, the glucose gets turned into glycogen as a storage. So you can sort of see how complex it all is. Signaling pathways, well, the nodes are typically proteins, and the edges are typically activation or inhibition of the downstream protein, often by phosphorylation of particular amino acids. These are, you can see, complex and interwoven, involves lots of protein-protein interaction, kinases, like this MAP kinase. Actually, kinase enzymes add phosphate groups to other proteins, etc. But it's not all proteins. This phosphoinositol 3 kinase phosphorylates small molecule on the membrane surface. So that's your more detailed insulin signaling pathway. Okay, so let's switch back to E. coli bacteria as an example of gene regulation networks. This is very much because E. coli is one of the best characterized organisms we have. So we can see in detail what the gene regulation network looks like. The human gene regulation network is not very well characterized and complicated by the fact that there are other regulation mechanisms like chromosome restructuring. But E. coli is quite a simple network. You have transcription factors. So these are the internal nodes representing genes which code for transcription factor proteins, which then bind onto other genes and either activate them or repress them. They either increase the gene expression of these other genes or they decrease the gene expression. So we have transcription factors as internal nodes, and then the links represent regulatory interactions. This transcription factor activates or represses this other gene, which is also a transcription factor, which activates or represses this gene, which, because it's a leaf node, it's probably a gene that does something. It's an enzyme that sort of causes the processing of a metabolite, small molecule, or it's a structural protein. It might be part of the intracellular structure of the cell. So this looks like a spaghetti mess of genes. How can we make sense of this? Well, if we use a program like Cytoscape, which is a a network visualization program, and we show it in the hierarchical format where these genes can only regulate genes which are below them, then we end up with this 5-layer hierarchy for the E. coli gene regulation network. And it's interesting because at the bottom we have all of the enzymes and genes which are building the protein. These are the final things that need to be turned on or off to do particular functions in the cell. And then we have different layers of transcription factors controlling them, with the highest up layer being master regulators. So one of them, CRP protein, decides whether the bacteria is in an anabolic state or a catabolic state. Is it building larger molecules from smaller ones like glucose and amino acids? Is it building up proteins and building up sort of glycogen? Or is it breaking them down? Is it breaking down protein to get amino acids, or breaking down starch or glycogen to get glucose? So it's a sort of switch between anabolic and catabolic states. The reality here is this looks complex, but there's actually a lot more detail. If we take this network and we express it like this is now showing you the detail of those interactions. We've got the CRP protein here, or the CRP gene. So we've got the CRP gene here. This is representing this line, the DNA, and this is another gene, the DNA with the MTLR gene. And then the arrow at the end is representing the expression of this gene as a protein. And then we have arrows like this indicating activation of the another gene. So the FAR gene, CRP activates, or the bar indicating repression. So it actually represses itself, but it also activates itself in a different region of its DNA. And this is some of the complexity. It depends on other situations, whether it activates or represses. Potentially the concentration, these might be different binding affinities. You can see a CRP here, it actually binds to 5 positions along the DNA of MTLR to activate it. But we have other genes which, for instance, the FAR gene represses itself in this one position, and this one, MAL1, represses itself in 2 positions along the DNA. And then there are complicated ones like GLNG, which both repress itself at this position and activate at this other position, but in this case it represses and activates. So this will be depending on the context of other things that are happening at this regulation site of this gene. So you can sort of see all of the complexity. You have the DNA, and then we're getting things like this binding onto it to actually repress and activate the expression of the gene. So you have to imagine the complexity of the previous diagram, but at this level of detail. And even this is abstract, schematic. We don't know, for instance here, the binding coefficients. What concentration does CRP need to be as a protein to actually bind and repress or bind and activate? So there is lots of detail even in a very simple organism like E. coli. So going into even more detail, there's this video on the Moodle site, which I recommend that you watch, which shows the molecular level detail of gene regulation. It actually shows the DNA molecule as an animation, this double helix molecule, and different proteins binding onto it in real time and activating. So it's a whole complex of proteins bind to activate the expression of the gene. So it's very worth watching. Okay, so turning back to the insulin response in humans to see how metabolic pathways work. So we had the insulin binding to the insulin receptor and then activating the signaling cascade, and eventually this leads to particular genes inside the nucleus and gene expression, gene regulation. And what does this finally do? Well, it turns on and off particular enzymes. So the ones indicated in blue here are being stimulated by insulin. So we've got glucose coming in, and this is glucose kinase. It's a particular enzyme that adds phosphate to glucose. Any kinase enzyme tends to add phosphate to something. So we've got glucose being converted to glucose 6-phosphate. And then we have an enzyme pathway consisting of enzymes like this PFK and PK, pyruvate, which takes the glucose 6-phosphate all the way up this pathway to the mitochondria, where basically they enter, they're consumed to produce energy. So that might be muscle being activated by insulin to absorb, to use up the glucose as energy. But we also have the turning on of glycogen synthase, which turns the glucose to glycogen as a storage protein. What other enzymes does this signaling turn off? Well, the ones in red are inhibited by insulin. Essentially all of the enzymes that are going in the opposite direction, turning pyruvate into glucose 6-phosphate, because we're trying to absorb the glucose and then store it or use it for different processes. So that's how the gene regulation links with the metabolic pathway, and the metabolic pathway is this small molecules being converted to other small molecules by different enzymes being increased in expression or decreased in expression. Metabolic pathways are one of the best characterized pathways, types of pathways. They've been studied for decades, and we have a database like KEGG which shows in detail the metabolic pathway. So we've got glucose here, and we've got an enzyme indicated by the square box, and this is the EC number. David might have talked about that, enzyme commission number. And this enzyme converts glucose 6-phosphate, or glucose, into glucose 6-phosphate. And then this enzyme turns it into fructose 6-phosphate, and then fructose 1,6-phosphate, all the way down via different enzyme pathways, all the way to pyruvate again. So we've got pyruvate down here. So that's essentially that pathway that we just saw in the liver cell cartoon. This is all of the detail stored in the KEGG pathway. And all of this, so we've got the nodes here are metabolites, small molecules, and the edges are labelled with enzymes to convert one metabolite to another metabolite. So this is all the detail, and this is all machine readable. The final type of key network is protein-protein interaction network, P2P. This isn't really a pathway, as in it doesn't represent biological mechanism like the signaling pathway or the gene regulation pathway or the metabolic pathway. This is just representing which proteins bind onto which other proteins. So it's actually because the experimental techniques allow us to detect proteins binding to proteins. This binding is used in all of the other pathways, signaling pathways, gene regulation pathways, and metabolic pathways. How is it used? Well, for signaling pathways, this might be a kinase which is binding to another protein to phosphorylate it, or it might be a protein binding to actually take off a phosphate. It might be allosteric activation of a transcription factor. So this protein binds onto a transcription factor to change its shape, to actually activate it to bind to DNA to express a protein, or an enzyme to activate the enzyme to process a metabolite. It might be inhibitory binding. This is the active site that turns glucose into glucose 6-phosphate, and this other protein binds to cover up the active site and act as an inhibitor of that metabolic process. It might just be simply physical closeness, that the glucose turns into glucose 6-phosphate, and it's very physically close to the next enzyme on the pathway, which may turn it into fructose 6-phosphate, for instance. So these are the reasons why proteins bind onto other proteins. And here's an indication of such a network. This time it's in yeast. It's the yeast sort of protein-protein interaction network, which was one of the first big networks which we sort of determined, and it was actually the actual technique being used is called yeast-2 hybrid, because it's actually originally developed in yeast. So in this case, the proteins, the nodes are proteins. All of these individual nodes are proteins, and the edges indicate a physical interaction between the proteins. So there's lots of properties of this network. It's thought to be scale-free and robust, so there's very few fragile nodes which have lots of interactions. So if you take out that one, you probably damage the system quite a lot because of all the interactions. But there's lots of nodes with very few interactions, so it makes the whole overall system, the whole network, robust. All of the red ones indicated here are where deletions are lethal. Actually, you can see this one isn't a lethal deletion, the big central node there. So it also provides indication of functional modules, that these are probably all interacting as a functional unit, the ones which are close together. So this protein-protein interaction is also used for protein function prediction. So where do we find out information about all of these biological networks? Well, each year the Nucleic Acid Research journal produces a database edition which actually describes hundreds of different databases in bioinformatics, and they form this compendium website that lists all of the databases over the last 10 years or more, and it's got descriptions of nucleic acid databases, protein sequence databases, structure databases, genomic databases. Each one of these opens up to hundreds of different databases. In particular, they have a link for metabolic and signaling pathways, and you can open up enzyme databases, metabolic pathways. I've opened up signaling pathways, and you have a long list. It goes well beyond the bottom of the page of all of the different databases which are present in their articles, and each one links to a research article about the database, so you can find out how it works, as well as a link to the database so that you can actually get data out of it. So that's a very useful resource, not just for biological networks, but actually for all bioinformatics databases. If you need to find a database on, I don't know, immunoglobulin folds, you can find under probably structured databases, and you'll find PDB, for instance, under there. Another resource which is actually dedicated just to network and pathway databases is PathGuide. So this is a resource which contains information about hundreds of different networks and pathways. They're under different categories, so it starts with protein-protein interaction databases, and you can see that there's a lot of them, and it just carries on down past the pages. There are hundreds of resources, and if you follow it down, you even have biological resources such as gene ontology, which David would have talked about in terms of function prediction, that these provide terms about biological function, biological process, and cellular location, and they're related by a directed acyclic graph. So they have a graph structure. They're actually in this pathway network database sort of compendium. And if going further down, we come to KEGG. So that's the metabolic pathway database that we were looking at, the Kyoto Encyclopedia of Genes and Genomes. There's lots of detail about each database, whether it's free or if it's actually needs academic registration, and also formats. Is it in SBML, Systems Biology Markup Language, or BioPax, which is an ontology-based format, which we'll discuss later. The formats of network data tend to be much more complex than sequence data. So there isn't really an equivalent of a FASTA file with just identifier and then sequence data. Networks consist of particular entries which are linked together. So there are many standards employing different XML languages. Systems Biology Markup Language. This is a format of molecular interactions, and BioPax, which we'll talk about later. So you can see this is actually the KEGG, an output from the KEGG glycolysis pathway that we were looking at earlier. So we've got glucose being converted into pyruvate, and if there's details of how the layout works in this particular case with XY positions of different entries. So these all have different formats. We'll discuss data representation of biological data in lecture 3 actually in more detail. So we'll end section 2 at that point, and we're going to move on from the network data onto omics data in the next section. Okay, see you, bye.